Great, thank you. I appreciate the invitation to talk this morning and everybody's patience as I try to close the session down. So at the Pacific Northwest National Laboratory, we have benefited from about 30 years of investments by the Department of Energy and NIH in developing mass spectrometry-based tools for biomolecule identification. So I'm going to talk you through some of the examples of the work that we've done and then focus a little bit on new technologies that are currently in development that may be useful for this question in the population. So you've heard a lot of talks about biomarkers over the last couple of days, but I want to reiterate that biomarker discovery really is a multidisciplinary contact sport. It requires biology and medicine. It requires your technologies to measure the different molecules you're interested in, and a lot of computation and data science to understand and make sense of the data with statistical models. And we also do a fair amount of imaging so that we can understand where the biomolecules are coming from, and that helps in the interpretation of the biology. So as we have developed these capabilities at the National Laboratory, we've been applying them to a lot of different areas. Our DOE mission space is ecosystem science and microbiome science, but we bridge that into human microbiomes, cancer, precision medicine, infectious disease, and exposure science. So in the case of exposure and military exposures, one of the things we need to think about to bridge between human exposure and precision medicine is really characterizing whether your biomarker is an exposure biomarker or a response biomarker. We can do diagnosis of health conditions. We can look at prognosis and risk stratification of different groups. We can look at mechanisms of response that could lead to therapeutic strategies. We can even do treatment stratification to figure out which people will respond to the best treatments, and then ultimately designing new therapeutics and new countermeasure strategies, whether it's treating or preventing exposure or response. So for example, some work that we did years ago, just using genomics-based technologies to understand carcinogenic potential of PAHs in the lung and in the skin. We did genomic studies looking at mice that were exposed to PAHs themselves, as well as some mixtures that contain PAHs, such as coal tar and cigarette smoke. And doing the animal study to actually measure tumor development, we could identify genomic signatures that are predictive of the carcinogenic potential. This data is collected after 24 hours of exposure to the animal, but then animals were followed out for a long time course of weeks to characterize their tumor development. And one thing you can see in the data is that although you get the same kinds of tumors from some of these compounds and mixtures, you get different levels, different magnitude, and different time frames, but we can use the genomic data to figure out which pathways are involved with the different chemicals and where perhaps they intersect biologically in their mechanisms. We've also done analysis of bronchoalveolar lavage fluid for proteins and lipids as a basis of understanding mechanistically what a genetic basis of respiratory failure was in infants. There's a mutation that's very well known. This ABCA3 is known to be associated with respiratory failure in newborns, but the mechanism wasn't very well understood. But doing proteomics and lipidomics analysis of the bowel fluid gave us the mechanistic linkage of this mutation to markers of vascular leak as well as markers of inflammation, so that we could determine really what mechanistically is going on in these infants and how you might prevent this or treat it early on. So some of the specific considerations that we heard about yesterday for this population really is that we have both acute and chronic health effects of chemical and particulate exposures. You have multiple co-exposures, including infectious agents that are seen in the in-theater environments. There's also a very large effect of genetics on exposure and response, and so we have to take that into account. And then, as again was mentioned yesterday, confounding impacts of lifestyle, stress, fatigue, and mental health all can exacerbate the response that you would get from these exposures. So the approach that we've been taking is really trying to capture a systems level analysis so that we can get the mechanism and at the phenotype. Exposures that you get from chemicals and particulates are going to hit at every level, as well as the metagenome, the microbiome that contributes to these things. And so what we try to do in our analyses is capture the gene expressions, the proteins, modifications, as well as small molecules and lipids that can be measured with mass spectrometry. So I'll give a couple of examples here, some work that we're doing in the field of type 1 diabetes. We've been working with the TEDdy cohort that follows young children who are at high risk of developing type 1 diabetes based on genetic or family associations. These patients are followed over time, and so there are several blood samples that are collected over time, both before and after the development of symptoms. And analysis of these data statistically and with the longitudinal sampling can provide us with statistical models that give pretty good performance on the prediction of which children will develop type 1 diabetes prior to the onset of clinical symptoms that are identified. And then we can use our statistical models to identify clinical markers that could be used for following these patients and be able to anticipate and follow their progression. We've developed an extraction protocol for samples so that we can do a single sample integrative analysis of proteomics, metabolomics, and lipidomics in parallel. And I've tried on most of my slides to include the references for you to look up if you're interested in more information later. With this method, we've been applying it to a lot of different studies where we can pull and collect these data in parallel from a single sample for individuals. So, for example, we did a study from Ebola-infected patients from the 2014 outbreak in West Africa. We compared those that survived, where we collected several samples, to those who succumbed to the infections, and compared to healthy people in the same area. And with the analysis of the genomics, proteomics, and metabolomics and lipidomics, we could identify a profile that would perfectly predict who would succumb to this infection from the very first blood sample that was collected in the Ebola treatment unit. And we can identify this as a signature that we would develop into a field deployable kit, for example, that could be used for triage in a location where resources are limited and you can't treat all patients with all methods. You could treat those that are the most likely to succumb. So now I'll walk through some of the advanced technologies that we're developing for biomarker discovery and how those might be used. Again, we're looking at spatial resolution of these markers and their expression in a tissue, getting down to single cells of resolution so we can get down to very small sample sizes. It's not really single cell, it's more like 10 to 15 cells, but it gets into some high resolution identification, high throughput specialized separations so that we can analyze potentially thousands of samples for known markers, high resolution detection and accuracy, and then really eventually moving into what we would call a standards-free global profiling of small molecules. So Nanopots is a technology that we developed and published this past year as a protein imaging tool that can identify more than 2,000 proteins with 100 micron spatial resolution. These images show where we've been applying this to a uterine epithelial project where there's some interest in understanding the difference between the markers in the luminal epithelium from stroma, because the lumen is the contact point for early blastocyst implantation. And we're interested to understand what may be unique in the cases where the implantation fails. And so we've been able to basically voxelate these slides of the tissue and through the proteomics technology be able to identify specific markers in like subsequent slices. For example, these are replicates here and here, and be able to identify specific markers of these specific regions. We're also doing a multiplexed selective reaction monitoring approach that allows us to multiplex, in this case, 146 peptide markers that were pre-selected and quantitatively measured in head and neck squamous cell carcinoma patients from the DoD serum repository. This cohort had 4 different time points of serum collection. So we had essentially 2 time points pre-diagnosis and 2 post-diagnosis. And with the analysis of nearly 1,000 sera samples, we were able to come down to a 13 protein classifier with an AUC of 0.9 to predict early detection of head and neck squamous cell carcinoma in these patients. We're also developing techniques for exhaled breath analysis so that we can detect biomarkers without having to collect more invasive tissues like the bowel fluids. And we've demonstrated that we can identify markers from the trachea, the lung, and the esophagus from exhaled breath. This figure shows the distribution of origin of the different markers that have been identified in exhaled breath. We can also identify volatile organic compounds that are being exhaled, as well as identifying markers of lung microbiomes. And then really the next step for us, it's like the next human genome project, is going beyond the reliance on reference libraries so that we can use chemical approaches or computational approaches for chemical identification. So if we think about the molecular universe, everything that's out there, we're really only measuring the known compounds, and very small number of those have standards that we can purchase. So upwards of hundreds of thousands of chemicals that we could potentially be looking for. It's very expensive and very slow to try to develop standards and create standard curves for all of them. And so reference libraries will really never be sufficient. And so the future, as we've been working on it, is in developing computational approaches that would utilize a third dimension of data that we can capture from ion mobility mass spectrometry to get at better identification, as opposed to mass identification, which is done with the 2D, and then be able to score the identifications and have some confidence in what you're identifying. So as an example, some of the work that we've done, we can actually, with the ion mobility mass spectrometry and collisional cross-section as the third dimension of data, we can separate and identify cis-trans isomers of the same compound accurately. What this figure shows that's hard to see is that in blue is the experimental collisional cross-section measurement, and in red is that which we predicted computationally. So we're validating these methods with experimental data. And in the case of the NTAC study, which was mentioned earlier, we can distinguish between 2 identical mass compounds that are different structures, one that was a true positive versus one that was a true negative, with about 91% accuracy in that study. And one of the things that we did do after the samples were unblinded is we used the true identifications as a way to enhance our scoring algorithm so that we could reduce our false positives below 10%. Again, pioglitazone was another one that was identified from the NTAC study where experimentally we had 4 data points that suggested different isoforms that were in the sample, and we could accurately identify that compound, purely computationally. So where we're going with this now is we've been working with partners in the Air Force and Department of Defense to think about how they could deploy simultaneous measures of physiological stress with exposure and response. We're working with LA firefighters who are wearing these physiological measures, collecting the telemetry data when they are in the firefighting environment, as you can see here. A lot of stress, a lot of fatigue, heat stress, chemical stress, inhalation. If we have the real-time data from them when they are in the field, if they're wearing wristbands that can be used to capture chemicals that are being exposed to externally, and then if we have plasma or urine samples that we can use to identify response metrics, we can get those from essentially kind of a real-time experiment of what's going on in the field at that moment. We have been working with Kim Anderson at Oregon State University with her wristbands, and as was mentioned, her standardized methods for extraction, she can now quantitatively measure more than 1,400 chemicals from a wristband. So just to summarize, and based on the work that we've done, single biomarkers are really unlikely to be kind of the gold standard of any diagnostic. So advanced technologies and data integration are really becoming more and more important. Response outcomes are really multifactorial and must consider genetic and psychosocial confounders that are going on in order to really reliably be able to get the precision for these panels to be used. And then real-time or parallel exposure measurements while the soldiers are in the field are really going to be imperative for understanding what is there at the time so that we know what to look for in the samples that we're measuring. And we can design biomarker panels for diagnosis, prognosis, or countermeasures. It really is dependent on the experimental design, the number of samples that can be collected, longitudinal sampling, and well-defined pathologies. So, thank you.
Well, thank you. Very, very interesting. David, did you have a question?
Yes. Given Mr. Patterson's talk a little bit earlier about the huge challenges in doing real-time measurements in the field, how would you implement, or how do you think it might be possible to implement your proposal for real-time measurements to get the kind of data that you need for biomarker work?
So I think today, with what is available, the silicone wristband or other kind of mobile technologies that can be worn with the soldiers in the field are probably about the best we can do to get a sense of what is in their environment at the moment and what we should be looking for. Again, those will always be challenged by measuring what you're looking for in a reference library as opposed to identifying everything that's out there. And so the global approaches will be more important in the future to figure out what else might be there that we didn't know we should be looking for. Ideally in the future, having wearable sensors on the skin that can sample for very specific markers that you're concerned about or looking for would be the ideal situation once we've identified what those markers are for those exposures. Whether the concern is a particular chemical exposure or whether the concern is acute toxicity in the field that you need to interrupt the deployment and get the soldier home really quickly because they're metabolically starting to have some symptoms of shutting down, kind of depends on the question. But quantum computing capabilities that are being developed right now will really enable the gathering of that kind of data from wearable sensors, and combined with artificial intelligence algorithms can help interpret that data coming in real time to make actionable decisions. So I could see it happening in the future.
Other questions? I'm afraid the hour is getting late, and the panel has enjoyed listening, and I think we're going to go ahead and say thanks.